Okay, so let's continue. Along the same topic of RNA splicing, scientists have discovered that something called alternative splicing is what happens inside eukaryotic cells. They find that not all exons needs to be recombined to produce just one mRNA for that one protein. But different combinations of exons can be cut out as well to produce different combinations of messenger RNA and thus different proteins. What I mean by that is you would think that exon 1 here with this DNA molecule template, you have exon 1, exon 2, and exon 3, and you think all of these, both of these introns will be excised, and at the end you have all 3 exons within the mRNA transcript, the mature mRNA transcript. But sometimes you can actually have different combinations where here you have illustrated where the exon 2 along with intron 1 and intron 2 was excised and degraded. Exon 1 and exon 3 are recombined instead. And more so, okay, there's another possibility where you can have exon 1, okay, and exon 2 recombined together, and what's looped out instead is exon 3. So this is another possible combination. So of course, the more exons you have, the more possible number of recombination, and you can have different proteins that's generated just by using one DNA template. So this gives an explanation why we have such diversity in the type of proteins we can produce, but yet only a small amount of our genome is encoded into proteins. It turns out that almost 60% of all human genes are alternatively spliced, but still, we are not entirely clear about how this process is directed or even how exactly the spliceosome does its job. We just know that spliceosome exists and is important for RNA splicing, and that this type of alternative splicing does happen in eukaryotic cells to produce different versions of proteins. There's still a lot out there for us to learn, even though we are years away from the discovery in the structure of DNA. As your textbook has discussed, the more we learn about how our genes and DNA works, the more complex we seem to find the processes are. We would think it works straightforward this way, just like how Crick thought all genes and proteins were collinear, and that, the number of nucleotides are always proportional to the number of amino acids. But diving deeper into the very details of each processes always tells us something else is going on. And it, in the end, it tells us that biology is not that simple. This is also the reason why I am always so fascinated about genetics. Okay, so we will end this chapter by briefly discussing the other classes of RNA that we brought up at the beginning of the chapter. Remember the very first slide, we had a list of all those different classes of RNA molecules. Some of them we have already discussed. We discussed pre-mRNA, we discussed mRNA. So the one of them that which we would discuss again in the next chapter, chapter 11, when we talk about translation, is transfer RNA, or known as tRNA. tRNA is responsible for bringing in that specific amino acid according to the codon sequences on the mRNA. Remember, in your mRNA molecule, you have those sequences, UAG, GGG, AUG, etc. Okay, and those every 3 nucleotide, we said that it was a codon. So what the transfer RNA will do is read those codons and bring in that particular amino acid that corresponds to the correct codon on the mRNA sequence. So tRNA contain a few rare modified bases that we don't find in DNA or mRNA molecules. For example, you have ribothyamine, pseudouridine, and inosine, and these are just a few. It also has a common specific secondary structure that is present in all transfer RNAs. It is called the clover leaf structure as depicted here. Okay, it looks like a little clover leaf. So this structure is formed by intramolecular hydrogen bonds. Remember those that connects the two strands of DNA molecules together. Those were hydrogen bonds as well. Okay, but they were in a sense intermolecular. Whereas here you have within that one strand of RNA molecule, you have intramolecular hydrogen bonds that will form and give you that clover leaf structure. Okay, so in order to match the correct amino acid with the specified codon, like I said, each tRNA has a specific anticodon that is complementary to the codons on the mRNA molecule. The 3' end of the tRNA is where the specified amino acid is attached. So this is the 5' end. This is the 3' end of the tRNA molecule. And you will have, for example, a big amino acid that is attached at that end. And here is where I was talking about where anticodon would be to bring in that particular type of or specific amino acid. And because anticodon is supposed to be complementary to your codon region, this is also made up of 3 nucleotides. And that's how transfer RNA is able to tell or make what sequence of amino acids according to the instruction that is listed or embedded within your messenger RNA molecule. Okay, again, just like your pre-mRNA molecule, the formation of tRNA process also involve transcription and followed by processing events that will trim and modify them to mature transfer RNA molecule, which we will not discuss here in this chapter. Okay, so the next classes of RNA we will discuss has to do with what we call gene silencing. This is a very unique tool we are now using in research to study phenotypes of different genes. Scientists have discovered and used these classes of RNA in research to study the effects of turning on or turning off a gene that affects whether the protein synthesis happens or not. So these two of them are called small interfering RNA, known as siRNA, or microRNA, which can originate from the cell itself, extraneously through extracellular fluid, or through a virus. They will combine with proteins, so proteins along here, to form a RISC complex, okay, which stands for RNA-induced silencing complex. This then can degrade your final mRNA molecule or represses the translation of that mRNA molecule. Both cases, whether siRNA or microRNA, will result in silence of a gene called RNA interference. So that's what the process is called or technique. So the sequence of these siRNA and microRNA is what tells them on which gene are they going to act on. So if this specific microRNA or siRNA is specific for insulin, then it will target the specific sequence for the insulin gene, and it will degrade that particular insulin mRNA molecule, therefore stopping the production of insulin just by adding either siRNA or microRNA. The third part or third type of RNA that participates in some sort of RNA interference is called CRISPR RNA. This is the last type of RNA we will mention from this chapter. It was actually initially discovered in bacteria quite a while ago, but its potential in genetic research has not been recognized until recent years. So these RNA molecules are encoded within bacterial genomes, and they have the capacity to cleave targeted sequences of foreign DNA. For example, those of bacteriophages, remember bacteriophages are viruses that specifically attacks bacterial cells. And just like any other cell, it doesn't like to be excuse me, attacked by viruses. So this was a mechanism that bacteria cells have evolved to prevent viral infection. So how this works is when a bacterial cell is first exposed to a foreign piece of DNA, let's say from a bacteriophage, it is then inserted into its CRISPR array. So that's what that's called. This whole thing is called the CRISPR array. So now this CRISPR array has this particular sequence that corresponds to your phage DNA molecule. Okay, so for example, those of, let's say, let me backtrack. So now, once you have that particular phage DNA incorporated into the CRISPR RNA, mature CRISPR RNA will combine with a protein called Cas protein. So it will be, the CRISPR RNA will be transcribed here to give you just the CRISPR RNA. CRISPR RNA will then combine with your Cas proteins to form what's called the effector complex. Now, it is like a sniper killer within the bacterial cells, ready for action because its target has now been locked in using that particular CRISPR RNA sequence. It is where we will look for that particular sequence within the bacteriophage, for example. So the next time that it sees the same foreign DNA molecule, again in the bacterial cell, the effector complex as a whole will now bind using the complementary sequence right here on the CRISPR RNA. Cas protein will then cleave the target foreign DNA, rendering it ineffective in invasion of the bacterial cell. Remember that the way a virus infects a cell is by inserting its DNA or RNA into a cell or host cell, in this case a bacterial cell, and multiplies itself using the bacteria's replication machinery because viruses don't have those machineries. If we can just get rid of their DNA to begin with, as soon as they insert their DNA sequence, then we will never have multiplication or replication of those virus particles. This technology of using CRISPR, which stands for clustered, regularly interspaced short palindromic repeats, has been actively used in research to easily turn off a specific gene of interest. And as I was making this lecture, I thought about how this technique actually can be used to target the scare of our current pandemic situation, COVID-19. When I did a search online, here is what I found. Development of CRISPR as prophylactic strategy to combat novel coronavirus and influenza. So there is already a group of scientists working on this approach to destroy this specific virus, because the goal of CRISPR and Cas protein is to get rid of and kill that specific foreign viral DNA. So hence, since we already know the sequence of the viral genome of the scary COVID-19 that's killing a lot of people and infecting a lot of people, we can essentially eradicate them from our community. So, of course, targeting and testing it in cells will be easy in vitro, meaning in a lab setting. But the challenging part of any type of pharmacology research is, especially in terms of CRISPR, even when scientists are thinking about using it to combat other diseases, doesn't have to be COVID-19. The challenging part is delivery of the proteins. We humans don't make Cas proteins, nor do we make CRISPR RNAs. So how can we deliver both the CRISPR RNA and the Cas protein successfully into human cells without them being degraded by our cells? It is always the problem of delivery whenever we're talking about production of a new medication or a new potential drug that will work for a disease. So regardless, CRISPR is definitely a very exciting field of genetics these recent years, and it has a lot of potentials, including eradicating a lot of single gene mutations like sickle cell anemia or muscular dystrophy that is just a switch of one nucleotide that can turn someone's life around due to genetic diseases. So this is something interesting we will conclude chapter 10 with. So let's continue our protein synthesis journey in chapter 11. See you. Oh, and the practice questions will actually be given in a different way, so it will be more interactive on eCampus instead of me just reading through the questions and going over the answers with you. So be sure to get to those practice questions once you're done learning the material. I would highly suggest for you to use those questions as a way to test your understanding of the lecture.